Today, I have the distinct privilege of introducing Dr. Ana Maria Cuervo. After completing her PhD and MD in biochemistry and molecular biology at the University of Valencia in Spain, she went on to do postdoctoral research in the Department of Physiology at Tufts University. She is currently a distinguished professor in the Department of Developmental and Molecular Biology and Medicine at the Albert Einstein College of Medicine in the Bronx, New York, as well as co-director of the Einstein Institute for Aging Studies. With more than 35 years of research in autophagy and lysosomal degradation in mammals, and over 30 years studying the biology of aging, Dr. Cuervo is an elected member of the National Academy of Sciences and a highly respected author, editor, advisor, and mentor. Please join me in welcoming Dr. Cuervo.
Thank you so much, Catherine, for that invite, and thank you so much, everybody, for giving me the opportunity of being there. Even I'm still in New York, so I'm sorry I couldn't make it in person. I would have loved to interact with all of you. But I hope you can see all my slides, right? And you can hear me, right?
Yes.
Okay, perfect. Yeah, so I thought I will give you some overview of what we are interested in the lab, and then try to have a couple of slides on some eye studies that we've been doing in collaboration. But please be gentle. I'm not expert on the eye, so I'm just trying my best here to get something that can be of interest. So as Catherine mentioned, we've been very interested in biology of aging. And the reason why we are interested is because, as you know, when we age, we have these 10, 15, now even 20 years of a gradual decline in health and function. And that's what worries us about aging. So ideally, we will go people to be like healthy, and then just drop dead. And in a way, that's not such a science fiction, because when you look at centenarians, and as some of you know, at Einstein near Barsila is doing all these studies with centenarians, this is really how they live. They are very healthy, and then they compress all the morbidity in a couple of months. And that has made the change, a change in the field, that we are not so interested of how long we live, but how long we are healthy. So the idea is to increase really health span and to make this curve to be flat. So all of us working in biology of aging are interested in identifying the cellular and molecular processes that contribute as drivers of aging, with the idea that maybe we can modulate them to get this extension in health span. And through the years, there have been different iterations of these hallmarks of aging or drivers of aging that are the ones that we all more or less agree. For our lab in particular, we are interested in two of them, the changes of the malfunction of autophagy as well as the loss of proteostasis. So I'm going to be mostly focusing in these two drivers and how we are trained to go about understanding why they drive aging and what can we do in order to restore their functionality to increase this health span. So proteostasis, as you know, refer to the fact that all the proteins, all the components inside the cell are in a very packed environment. So when you think in the cell, we always make it look like this fried egg. But when you look at the structural level, it's really a very, very dense environment. This is a 3D reconstitution. You can see here proteins, lipid droplets, ribosomes, and everything is very packed. So that makes absolutely necessary that each of the 1,000 of proteins that you have inside the cell is on the right conformation, at the right place, and at the right time. And in order to make sure that this happens, because otherwise you will have aggregation and unwanted interactions among these components, you have all the cells in your body have what is called the proteostasis network. And the major components are really the chaperones that are the ones identifying the conformation and the place where a protein is. And then you have the proteolytic systems. When the chaperones cannot restore the conformation of a protein or bring it to the right place, they can always eliminate them. And then there are two major systems, the ubiquitin proteasome system and then the lysosomes or autophagy. So the importance, as I say, of maintaining this proteostasis has been closely linked to disease. So there is a long list of what is called protein conformational disorders, and I just put here some examples of the ones that we've been interested in, which the protein that misbehave is different depending on the disease, but at the end of the day, this loss of conformation and this loss of proteostasis for these specific proteins and the ones that bring along result in aggregated oligomers, whatever species you want to name, that destroy functionality because they interfere with normal function, and in many cases results in cellular death and in the loss of function of the organ. So for most of my talk, I'm going to be focusing on neurodegeneration because it's a part that our lab has been working in, and then I will try to sprinkle some data that we have acquired in degeneration on the retina. And of the different components of the proteostasis network, as Catherine mentioned, my lab has been interested on autophagy. And probably autophagy doesn't need an introduction for this group, but just so we all start on the same step. So autophagy refers to the degradation of any kind of components, proteins, aggregates, organelles, lipid droplets by lysosomes. So lysosomes, as you know, are these acidic compartments that contain all kinds of hydrolases that can degrade any kind of intracellular components by this process of autophagy. So when we think about autophagy, one slide that I always like to incorporate is this one to illustrate that the cells can do autophagy by very different mechanisms. And this depends on how you take something from the cytosol and bring it to the lysosomes for degradation. So there is a mechanism that is conserved from yeast to mammals that is called macroautophagy. So when you hear autophagy, in most cases, this is the pathway that people are referring, where you basically trap wherever you want to degrade. Imagine it's an aggregate, and then you make this double membrane vesicle that has trapped your cargo, what you want to degrade, to fuse with the lysosome just to degrade these materials. And this process, as I mentioned, is conserved from yeast to mammals, and it comes in different flavors depending on what you are degrading. So you can talk about lipophagy that we described a long time ago, when you selectively degrade lipid droplets, mitophagy if you only degrade mitochondria, glycophagy, aggregophagy. But in all the cases, the mechanism is the same. You create this freestanding double membrane vesicle, and then what is different is the receptors of how you identify what you want to degrade at every time. But even this is the form of autophagy that has been more extensively studied. In the case of mammals, you also have the possibility to do other types. For example, you can just invaginate the membrane of the lysosome or the late endosome and trap little portions of the cytosol, proteins, but also fractions of organelles in this process that are known as microautophagy. And some of them are also conserved from yeast to mammals. But then in mammals, there is a third form of autophagy where you don't require formation of any kind of vesicles, as in macro and micro, and you directly transport proteins across the lysosomal membrane for degradation in the lumen. And this process is known as chaperone-mediated autophagy. And for the rest of my talk, I'm only going to be talking about this type. I'm more than happy to entertain questions about the others, but I wanted to keep it simple and focused, also because it's the one that we have dedicated more efforts in the lab. So this process is very simple. I will be referring to it as CMA for short, like chaperone-mediated autophagy, and it recognizes single proteins that contain a targeting motif. This motif was described by the late Fred Dice, and he found that proteins that have a motif biochemically related to this pentapeptide can be recognized by a chaperone, the constitutive member of the HSP70 family, this HSC70, that basically identify, when it binds to this motif, it identifies this as a protein that has to be degraded in lysosomes via this CMA. The motif, as most targeting motifs, is degenerated. So if you are interested in knowing if your favorite protein or your whole proteome, which proteins have this motif, we have put a free tool online that you can use to identify these motifs. And then the other key component, I mean, there are many more components of this pathway, but the other one that I want to highlight here is this single transmembrane protein, this lysosomal associated membrane protein type 2A, because it serves as a receptor for this pathway. The process is very simple. You identify, for example, this red protein, the chaperone identifies it, brings it to the tail of LAMP2A at the lysosomal membrane. The chaperone leaves, and then leave the substrate attached to this single transmembrane protein that then multimerize to form a translocation complex. And then you have a luminal form of HSC70 that pulls the protein, and the protein is degraded in the lumen. So as you see, it's very simple, and it occurs in a subpopulation of lysosomes inside all the cells of your body. So CMA is constitutively active, as I mentioned, in all the cells of your body as far as they have lysosomes, but it can be induced under conditions of stress, and this is examples of stressors, so it's part of the response of cells to any kind of a stressor. When it's activated, it has important functions. So some of them are related to quality control. So as I say, any protein that is misfolded or that is oxidative damage or is in other compartment, the same way that you can send it to the proteasome or other forms of autophagy, you can also send them to CMA. It's also important as a source of free amino acids. When you degrade these proteins, these free amino acids can then go back to the cytosol and utilize them for synthesis. And as I will try to illustrate through the talk, it's also important to terminate the function of proteins and have a regulatory role inside the cell. And I will tell you a little more about this regulatory function in a moment. So through the years, we've been very interested in developing tools to visualize this process. So originally we used really biochemistry, isolation of lysosomes and reconstitute it in vitro. But several years ago, we generate a fluorescent reporter that contains this targeting motif. And when it's delivered to lysosomes, it makes them fluoresce in green or whatever is the fluorescent that we use. So then if you have your protein, your substrate protein is diffused in the cytosol, that means very low activity for this pathway. But as you start seeing all these dots that corresponds to active lysosomes for this process, that means that there has been an activation of CMA. So Xuxian, a former postdoc in the lab, developed this transgenic mouse model that expressed this reporter in all the cells in their body. And in collaboration with Jose Javier Bravo Cordero, we started to use two-photon microscopy or just regular fluorescent microscopy in different organs to map how is CMA activity. So I'm showing you here, for example, this is the hippocampus of a mouse. This is a healthy, normal mouse. And as it gets close, you start seeing these little dots that corresponds to lysosomes where CMA is activated. And then you can start doing all this mapping. So for example, if we map the hippocampus of this animal, you can see regions like this CA1 that has this beautiful, very high levels of constitutive basal CMA. Meanwhile, other regions you have lower amount of CMA. And then, of course, you can go into single cells. So this will be a neuron. And then when we go inside, not only you see that there is CMA, but you can determine where in the cell this CMA is happening. So we've been using this animal to really understand a bit more about the physiology of CMA. So for example, Yves and Susmita in the lab, using this mouse model, identified that CMA undergoes circadian activities under the circadian control. So for example, if they were coming to the lab every 6 hours and isolating, in this case, I'm showing you liver, and then these are so this is, remember, these are mice, so they are nocturnal, and this is the day and this is the night. And as you can see here, as you go through the day and night, there are major changes in the activity of CMA. And what is interesting, we know is under the circadian clock, because if we eliminate a component of the clock machinery, BMAL, it's not that you don't have CMA, it's just it doesn't cycle anymore. And something that I found very interesting is that depending on the organ, as you look here, this is liver in blue, kidney in green, and heart in red. And in all the cases, you have this cycling, so CMA cycles during the day and the night. But if you look here, the peaks of maximal activity and minimal are different depending on the organ. And actually, we even did cell type specific, and different cells inside the same organ cycle at a different time. And then just to complete a little the study, not only we showed that CMA is under control of the clock, but also CMA degrades key components of the clock. And this is required for periodicity because you have transcriptional upregulation, but then somebody have to bring down these transcripts or these transcription factors in order to have a cycle of down and then another up again. So we found that if you don't have CMA, these factors remain very high and you have a disruption of most circadian processes in the body as well as disruption of the cycles. But as I said, this is an example of a new physiological function that we have found for CMA. And then another one that we published recently is changes in CMA, for example, during exercise. Everybody asks, how can you activate CMA? So the sad news is that you have to exercise. So if you compare here, this is work from Olaya. We are looking at the skeletal muscle. And remember, more dots means more activity. So if you look at females and males, resting conditions, as soon as they start exercising, and this is just like 20 minutes in a treadmill. And as you can see here, you have beautiful activation of CMA, both in males, but sadly enough, females benefit more. So I don't have an excuse to don't exercise anymore. But this is another example of physiological conditions in which CMA gets activated. And then, of course, through the years, we have also used these mouse models to try to understand in which pathologies there is disruption of CMA activity. And these are papers from our lab, but many other labs have also confirmed that CMA activity seems to be compromised in neurodegenerative conditions, immune disorders, metabolic conditions, and cardiovascular diseases, also in cancer, and in some eye-related disorders. So for the purpose of today, as I say, I'm going to be mostly talking about neurodegeneration, and then I will show you some examples of dysfunction of CMA in diseases of the eye that we have done in collaborations with other labs. So the main reason why we started this interest in neurodegeneration is because we realized that many of the proteins that have been associated to neurodegeneration, I have here some of the Parkinson, Alzheimer, Huntington, they contain these CMA targeting motifs. And that doesn't mean anything. That means that at some point in the cell, they can undergo degradation by CMA. The problem happens when you look at the pathogenic variants. And of course, we did this in collaboration with amazing neuroscientists. I'm not a neuroscientist, I'm just cell biologist playing with neurons. But what we found is that when you have the mutants or the pathogenic variants, the chaperone still identifies this protein as something that needs to be degraded, and it interacts with the targeting motif. It brings it to the surface of this lysosome, since as I showed you in the cartoon for the normal proteins, but unfortunately these proteins, these pathogenic variants, they get stuck at the lysosomal membrane. So this is bad because then these proteins cannot be degraded by this pathway, and we think that that contributes to proteotoxicity. But it gets worse because not only these proteins are not degraded, but also proteins that are normally degraded by CMA, now they cannot be degraded because these proteins end decorating the surface of the lysosomes, preventing the access of other substrates. So through the years, what we have proposed is that in all these neurodegenerative conditions where you have the pathogenic variants of these proteins, you have problems with the proteostasis of the protein that is the pathogenic one, but also you have an additional problem because of the malfunctioning of CMA. But through the years, again, we have done all these studies in vitro because we didn't have a mouse model until recently. So now that we have this mouse model where we have a reporter that we can follow CMA, the question was, this blockage that we've been reporting to happen in neurons in culture for many years, does it happen also in vivo? So we have used different mouse models, and I know there is not perfect model of neurodegeneration, but in this case, Aurora and Mathieu used one of these triple transgenic mice that express APP, presenilin 2, and also a pathogenic variant of tau, as you can see here. And we saw that that was enough, that when you measure CMA activity in neurons, so this is in normal control animals, and just by expressing these pathogenic proteins, the activity in the brains of these animals decreased dramatically. And this decrease occurs initially in neurons, and then it starts affecting microglia and astrocytes. And this is not something unique for frontotemporal dementia or Alzheimer or tau tauopathies, because Adrian did the same using one model that expressed this alpha-synuclein, another disease, what we have shown in vitro, this decrease on CMA, and sure enough, if you compare dopaminergic neurons in control and in the PD model, you have this dramatic decrease. And we have done this not only in the genetic model, but also using preformed fibers microinjected in the brain, and then you see also an inhibition of CMA. So this has convinced us that this decrease that we see in vitro, it also happens in vivo. And then the other thing that we have used this model is to see changes in CMA with age. So what we did is try to compare how CMA changes systemically in young and old animals, and also comparing males and females. And this was only possible because everybody in my lab knows about some organ. So we were able to analyze each of these organs and different cell types inside each of the organ. So the study was done at the single cell level, where we combine single cell imaging of each of these organs and the cell types, but also single cell RNA-seq, just to see or to discriminate in which cases CMA changes are transcriptional or not. So when we do the mapping of CMA in these organs, and this is just a young, healthy animal, what we can see is there are organs in which you have already very high levels of basal CMA, like the liver, the pancreas, and there are organs where you have these gradients, for example, in the brain or the retina, that is very dependent on the cell types. So the basal levels of CMA are very dependent on what is the cell type that you are looking on those organs. And that's to summarize, I mean, this was published early last year, and just to summarize a little what we learned. So we learned that the basal level of CMA activity is very cell type specific and organ specific. Then when we look at CMA activity in general, I know it's very bad to generalize in science, but in general, we see higher CMA activity in females than in males. So for example, if you look different parts of the brain, and we are looking here at females and males, and this is their level of CMA activity in different regions. So I'm showing you here, for example, C1 region in the hippocampus in neurons. You see how these young, healthy animals, in females, you have significantly higher CMA than in males. Then, of course, there are always exceptions. I don't want anybody to get demoralized. So if we do the same mapping in astrocytes, as you can see here, in astrocytes, basically, we don't see those differences, or if anything, in males, is a little higher. And then what happened with age? So again, in general, with age, we see a decrease in CMA activity, both in females and males. And again, there are some exceptions. For example, for astrocytes, we see a decrease in females, but in males, we see this increase that now we know is related to the non-cell autonomous regulation. But this is basically, as I said, we have done a map for almost every organ that we have expertise in, with the idea of learning how aging affects these organs, and also if we can start understanding vulnerability to disease based on the levels of activity and how they get affected in age. We also did some studies in the retina, and these are all in collaboration with Patricia Boya, and Raquel was the amazing postdoc that was in between both labs here in New York and in Spain. And basically what she did is first map in the retina different cell types, and what she found is that if you compare levels of CMA activity through the different layers of the retina, is clearly the outer nuclear layer, the one that has higher activity. So the photoreceptors displays the higher activity. And then when you look at the effect in aging, she was part also of this aging study. I'm showing you here in the top rods. In the bottom are cones. And again, you can see that there are some sex differences. So for example, males have higher CMA activity in rods, but not sex difference in cones. And then what we see with age is that the decrease is preferentially happening in males. Actually, at this age, so this was 24 months, in females, if anything goes up, there is a compensatory increase that then we see if we follow until 30 months of age, we see that decrease. But the kinetics between males and females, they seem to be different. And then we also tried to look in conditions of disease, also in the retina. So the first disease that we look at was retinitis pigmentosa. So what we notice is that if we use some of these mouse models that recapitulate some of the mutations of this retinitis pigmentosa, as most of you know, these are very aggressive models. But if we follow the changes of CMA in photoreceptors, you can see that there is this initial upregulation, and then there is a decline, and if you follow, eventually goes down. So we thought that this was interesting because we have seen that in other conditions, that in response to damage, you activate CMA, but then if the damage persists, or is very severe, eventually CMA gets even inhibited. So we thought that this, as I will show you at the end, could give us the idea that maybe if we are able to preserve this CMA upregulation during all the time of the disease, we might be able to preserve the retina. But the first thing that we wanted to know is like, okay, this is fine, but this is a mouse model. Does it happen also in patients? So, of course, in patients, we cannot use these reporters yet. So what we have to do is to try to use approximations by measuring components of CMA, or even better, measuring the whole network. So this is work from Mathieu and Adrian. And what they have done is that if you follow a single component of CMA, as I mentioned, I only talk about the chaperone and the receptor, but these are other regulators and modulators of this pathway. So if you follow one of them, you might see a correlation. But when you follow the whole network, and this has been shown computationally in many other settings, you have a much robust idea of how the activity of this pathway changed. And because we have the mice, we were able to compare and make sure that this algorithm that we have developed to calculate or predict CMA match, at least in mice, with the activity that we see. So the changes, the algorithm is very simple. What we did is gave weight to each of these components, depending on how important it is for the pathway. And we've been 30 years now studying this pathway, so we got some idea of who is important and who is more or less important. And also directionality. Are they activators or are they inhibitors? Because if you just use the go terms, they tell you autophagy or CMA, but they don't tell you if it goes up and down. So we felt that we needed to develop algorithms that at least help us to predict, is this going to be increased CMA or decreased CMA? So when we did that, and we test in many systems, so if we use the IPS derived from individuals with retinitis pigmentosa or not, if you go to the CMA index, you can see here that compared to control, so these cells from the control individuals or healthy individuals have this nice increase in CMA, and they start already with high activity. Meanwhile, the cells from the patients have lower predictive CMA activity, and they fail to really reach the maximal activity that you see in the adults. So this kind of gave us the idea that CMA activity decrease in retinitis pigmentosa, not only in these models, but also in the patients. And more recently, we also did some studies inducing degeneration. So, for example, if you use some of these degeneration models, this is chemically, Raquel showed that there is again this decrease in CMA activity. So any persistent stress or chronic stress is gonna decline, is gonna reduce the activity of this pathway. But we also wanted to know in age-related macular degeneration. So this is work from Iñaki, also in Patricia's lab in collaboration with Ted Farrington. And here what we did is to look at patients. So if we look, for example, at retinas from explants from the patients, we see that the higher or the lower CMA activity, or the most affected changes in CMA, in this case is in the retinal pigmental epithelium. So if you look here, the CMA score in these patients is clearly significantly decreased. And when you look at the major components, both the receptor at the lysosomal membrane as well as the chaperone seems to be significantly decreased in each of the patients that we analyzed, the explants. And then with Deb, she was able to differentiate IPS into retinal pigmentary epithelial cells. And as you can see here, those ones coming from the patients has a significant decrease in CMA activity. So this gave us some evidence that not only retinitis pigmentosa, but other conditions of degeneration, you have a compromise on CMA activity. And then just to go back to our neuronal models and neurodegeneration, I mentioned that we've been very interested in neurodegenerative conditions, but also in the aging on the healthy brain. So using this algorithm, we've been looking at data, and we can do it in single-cell RNA-seq or proteomics. And if you look, for example, these are brains from healthy individuals from 40 to 80 years old. And as you look at their CMA score, different regions of the brain, you see this gradual decrease with age. This decrease is much more accentuated when you have disease. So, for example, Mathieu used this single-cell RNA-seq that was already published of individuals with Alzheimer at different BRAC stages, so progressive disease. And because it was single-cell, now we can see what are the cells where CMA activity changed. And as you can see here, these are excitatory and inhibitory neurons. And you see that this gradual decrease that happened with age here is much more accentuated, and the decrease is higher as the disease progresses. And I know it's correlative data, but I was excited when we tried to correlate the level of remaining CMA activity with the level of pathology. In this case, the neuritic plaques, the beta plaques, you can see that the lower is your CMA activity, the higher is your plaque burden, and vice versa. So again, it's correlative, but this is in humans, but there seems to be this inverse relation between the robustness of your CMA activity and then the propensity and the severity of the disease. So basically, we wanted to know, okay, now I hope I have convinced you that CMA activity decrease in many organs with age, and also this decrease seems to be accentuated in age-related diseases, either neurodegenerative conditions or, for example, some of this macular degeneration that we see in the retina. So what are the consequences of this reduced CMA? So starting with the brain first, what we did is to try to see what is the major component that contributes to the decrease on CMA as we get old. And what we found is that independently of the organ, in almost all of them are the levels of this receptor, the ones that decrease very much. So for example, I'm showing you here 3 different ages, and then this is mice. And these are 3 different brain regions. And yeah, the dynamics might be different, but in all the cases, you end with lower levels of the receptor. And I can show you the same for the retina, the same for liver. Most of the organs, the problem is a decrease on these levels of the LAMP2A, sometimes transcriptional, but in most cases, it's a problem of stability of this protein at the lysosomal membrane. So we thought that because this is the most common effect that we see, this might be a very good tool to try to mimic the changes that we see with age in a young animal and see what happens. So this is work from Mathieu, and what he did is to selectively eliminate this receptor, this LAMP2A, either in GABAergic neurons or in dopaminergic neurons, and then see what happened to those animals. And when you look at them, I mean, he did all kind of behaviors, but depending on the neurons, the type of neuron, you see that there are signs of neurodegeneration. So this is the typical clasping. As the animals age, clasping start increasing, and we start seeing phenotypes by 6 months, 4 to 6 months of age. They have, for example, lower memory when we do the white maze compared to controls. Doesn't matter which type of neurons, or if it is total or if it's specific for neurons, you have this decline in memory. And you also have, for example, when you do the TH neurons, you can see a shortening of the stride. So basically, it's clear that these neurons are not functioning so well. We did a lot of electrophysiology, but also we look at these neurons, and what we saw in the brains is that if you compare the brain of controls or mice where we have just eliminated CMA in neurons, the tau, the alpha-synuclein protein that you see here, is the one that comes with the mouse. This is not mutant. Just by eliminating CMA, look the accumulation that you get of tau. Again, wild-type tau. And it looks like the controls almost don't have, because I want you to really appreciate the increase that we see by just blocking CMA in neurons. And then if you look at alpha-synuclein, the same, and you even get to see this formation of aggregates. So I want to pause here a little, because I'm not implying in any way that CMA degrades aggregates. CMA can only degrade single proteins because it requires unfolding in order to cross the lysosomal membrane. But what we think that CMA does is to control this metastable protein, this group of proteins that are very prone to aggregate as soon as they pass their saturation limit. And I always use the same example of the salt in the water. It's like as far as these proteins are at a low concentration, they are soluble and you don't even see any aggregation. Once you start having levels higher than their saturability index, then they start aggregating. So we think that when you don't have CMA, as it occurs in aging, or in some of the diseases that we talk, these protein levels are going to increase, and then you are going to promote aggregation. And just to put it a little more scientific than the water and the salt, if we look at the supersaturation score of the brains of control animals, or the animals that do not have CMA neurons, you already see the shift of the whole proteome to reach values closer to saturability. So we think that part of the problem when you don't have CMA in neurons is that even the other components of the proteostasis machinery are working. They seem to don't be able to compensate for the role that the CMA has in maintenance of this metastable proteome. A consequence that we saw of this is that once you have this blockage of CMA, there is a time that the cell has a way to partially compensate. So, for example, the same chaperone that I mentioned can bring these proteins to CMA, can also bring them, if CMA is blocked because of your pathogenic tau or because you are getting old, the same protein, the same chaperone can bring them instead of to lysosomes to this late endocytic compartment. So these are multivesicular bodies, they have proteases, and they have relatively low pH, not as low as lysosomes, so they will be able to degrade them, and that's the end of your problem. So initially you don't see major problems with proteotoxicity, and that's why the phenotype occurs past 6 months and older. However, as the animals get old, and all of us, we published recently that the degradative capacity of this compartment also decrease. So all of a sudden you are rerouting these proteins to this compartment, and now this compartment cannot degrade them anymore. And what we saw that happen is that if you cannot degrade it inside, that results in some molecular changes that makes this compartment now dock into the plasma membrane and release all this garbage outside. So in a way that gives a break to the neurons because now they are throwing the garbage outside. But in the long run, this is not going to be good because we show that this contributes to propagation of disease. And just to show you that this is not something that we just see in mice. So what we did is we were in collaboration with Allison and Lee Gunn, we were able to get 9 brains of Alzheimer individuals and healthy individuals, age and sex match. And I'm just showing you here one of each one. And if we isolate these late endosomes corresponds to this compartment, as you can see here in healthy individuals, you don't see the presence of this pathogenic tau going there. When you look in the patients, you see this pathogenic tau coming to these compartments, the same thing that we saw in the animals. So we think that this molecular signature is something also happening in the patients, and our goal is to prevent this blockage or revert this blockage of CMA to avoid these proteins to go outside and contribute to more proteotoxicity. But as I mentioned, part of the phenotype that we see in these animals is related to the role that CMA has in proteostasis, and this role is shared with other components of the proteostasis machinery. So if you don't have this pathway, you saw those aggregations. But in addition, something that we have become to realize through the years is that CMA is not only important for quality control, as not only degrade proteins because there are damage, but it can degrade fully functional proteins just to terminate whatever they are doing in the cell. So in a way, this is very similar to the proteasome. For example, as you know, if you want to progress through the cell cycle, you have to degrade cyclins by the proteasome at very specific times. So it turns out that CMA also contributes to that kind of very selectively and timely targeting of a variety of proteins, not because they are damaged, just just to terminate whatever they are doing. So through the years, again, my lab and other labs have identified many of these proteins. So for example, many proteins that participate in glycolysis or lipogenesis. So a lot of metabolisms can be regulated through CMA, but also transcription factors, components of the immune response, cell cycle, circadian, as I mentioned at the beginning. But also very recently, we found that CMA in neurons also contributes to modulate neurotransmission. So I wanted to just give you an example of how CMA goes about this regulatory role. Again, this is not related to proteins because they are damaged. This is just not removing proteins at a time that they have to be terminated or whatever they are doing. So this is work from Rabia, and the whole thing started working with this model where we don't have CMA in neurons. Something that the caretakers keep reporting is that these animals develop seizures, spontaneous seizures. So Rabia decided to do it in a more regulated manner and just induce seizures with one of these agents, the pentanyltetrazole, and basically see how it compares in a normal animal or an animal that does not have CMA either in the whole body or just in neurons. And as you can see here, if you compare the severity of the seizures, the time or the onset, the time for onset, or the survival of the animals, if you don't have CMA in neurons, I mean, in the whole body, obviously, you also have like more severe seizures, but just by not having it in neurons is enough to show the whole phenotype. And actually what we saw at the molecular level is that when you eliminate CMA in neurons, here we are doing microelectrode recordings in neurons in culture, and as you can see here, there is a phenotype of hyperexcitability. So not having CMA in neurons made the release being exacerbated and asynchronic. So basically you have this hyperexcitability that is the basis for these seizures. So Rabia wanted to know how is CMA doing this? Why is there is this hyperexcitability? And I'm not going to go in detail now, it's published, but basically she didn't find major structural changes, no changes in properties of the neuronal membrane, no changes in the ratio of excitatory to inhibitory neurons that we were considering for a while. But things got a little more interesting when she started looking at calcium dynamics. So the animals that do not have CMA neurons will always be in red. And as you can see here, when we look at the cytosol, they always have higher levels of calcium. But when you look at the ability of mitochondria and endoplasmic reticulum to buffer this calcium, they don't seem to be doing a good job. So basically, CMA somehow is modulating calcium-related proteins and calcium dynamics, and that probably contributes to this aberrant neurotransmission. So then in collaboration with Pablo Castillo, we wanted to know at what level is this presynaptic neurotransmission or postsynaptic? And basically what we found here is that if you do recordings, for example, with presynaptic protocols, we see different phenotypes if we look at females or males. So when you look at the females, as you can see here in red, the ones that don't have CMA have higher excitability, presynaptic excitability. Meanwhile, the males there were some significant changes, but they were not as dramatic as we saw in the females. However, when we look at the postsynaptic, the picture was quite different. So if we look at the postsynaptic here, the females don't have major changes, but then the males are the ones that have this postsynaptic excitability. So this kind of become interesting because it's like, okay, depending on the sex, in both cases we have seizures, but in one case there are presynaptic and the other are postsynaptic. So we wanted to know, are they degraded the same through CMA? So what we did is to do comparative proteomics. These are brain slices, and basically what we did is compare animals that have CMA, animals that do not have CMA, and then analyze CMA substrates. So these are proteins degraded in lysosomes in a LAMP2A dependent manner. You just have to trust me on this, but if we plot, for example, these are males, and these will be females, and these are CMA substrates. So you have some CMA substrates, the one in pink, that are degraded both in males and in females. But then you have this group of proteins that are only degraded in females, and this group of synaptic proteins that are only degraded in males. And this is completely independent of the abundance. It's not that these are more abundant in females and these are more abundant in males. Completely irrelevant. And then when we did pathway analysis, it becomes pretty interesting because when you look at the CMA substrates in females, all of them seem to be more in the presynaptic region. Meanwhile, the ones that are unique in males seems to be postsynaptic. And if you remember, that coincides with our electrophysiology. So the females, where you have more remodeling due to CMA, are the ones that when you don't have CMA have a presynaptic hyperexcitability, and the males, where CMA takes care most of the remodeling of the postsynaptic proteome, are the ones that when you don't have CMA have more postsynaptic hyperexcitability. So we think that the sex differences is not only quantitative, females have more CMA than males, but also females and males are degrading different proteins at a given time. So then this was for the neurons, but what do we know when we don't have CMA in the retina? I showed you that with age, there are some specific cell types in the retina where CMA activity decrease, but also that in macular degeneration, retinitis pigmentosa, you have this decrease of CMA. So is this something that might contribute to the phenotypes or to loss of function? So Raquel actually generated this animal again where she's selectively eliminating LAMP2A, so no LAMP2B or LAMP2C, so just eliminating CMA. But we are using a systemic model because we wanted to see all the different cell types in the neuron. And what she did is measure visual function, and these animals by 8 months of age, they start developing these significant differences. So the purples are the knockouts. So basically, if you don't have CMA, you have a loss of visual function. And when you look at the thickness, again, the purples are the ones that do not have CMA. So there is thinning of the retina. And as you look at here, you have a loss of rods, but also shortening of the cones. And then when we look at the cellular level, it's like, what do we see there? So the animals that do not have CMA, when you look at the retinas, they have an increase in cellular death. So this is tunnel. They have higher proteotoxicity, so we are using proteostat, but we also did it with ubiquitin, and you kind of see the same aggregates that we were seeing in the neurons, if you remember. And also they have gliosis, so they have an inflammatory phenotype. So this is IBA1, very dramatic increase, but also GFAP for astrocytes also increase. So basically just the loss of CMA with no other stress added seems to be enough to result in some level of functional loss and eventually cellular loss in the retinas. So we wanted to know a bit the mechanism, and of course, as I showed you in the proteostat, it's very clear that CMA contributes to maintenance of proteostasis. When you don't have CMA, you start seeing the accumulation of these aggregates the same way that I showed you that happen in neurons. However, we also thought that because in neurons we have seen these regulatory functions, it might be interesting to know if part of the phenotype that we see is also related to loss of function because you lose these regulatory roles of CMA. So we did very similar to what I showed you just with rabies neurons. We took retinas from the CMA competent and incompetent retinas, and then we did degradation studies so we can really identify the degradome, the pool of proteins degraded by CMA. And when we do that, you see accumulation of a bunch of proteins. And if we do pathway analysis, you find that the major dysregulation is related to metabolism in the retina. And actually we did metabolomics and we found that just eliminating CMA changed very much the glycolytic pathways as well as the OXPHOS pathways in the retina. And the other one that was very interesting, we found alterations in the proteasome. And these are primary alterations because what we found is that many subunits of the proteasome undergo degradation by CMA. So if you don't have this pathway, you change the stoichiometry of the proteasomes and they become less functional. So part of this proteotoxicity that we saw, in part is due to CMA loss, because that's what we did, but in part is because CMA cannot modulate this proteasome assembly properly now. You also contribute this increased proteotoxicity. So again, it's a combination of quality control, but also regulatory roles of CMA in the retina. So then what can we do about that? So the last couple of slides, I just wanted to tell you about things that we are doing, trying to fix CMA, because not all of us might develop macular degeneration or Alzheimer, but definitely all of us are going to get old and the levels of LAMP2A and CMA are going to decrease. So what we have done is to engage in a 3 front approach. We are using a genetic model where we overexpress this receptor, the one that decreases with age, and then we see at what happens if you restore or prevent this decline of CMA with age. We are also doing some dietary interventions, but for the sake of time, I'm going to focus on what we have done with drug development. So this will be pharmacological activation of CMA. So we have done this work in collaboration with Evris Gabathiotis because he's an amazing medicinal chemist. And something that we found through the years is that there are endogenous inhibitors of this pathway, such as the retinoic acid receptor alpha, that maybe we can selectively modulate to take out that brake and activate CMA. So Evris developed some molecules that are not acting on the catalytic site of this receptor. They are acting on this anchor, a repressor of this receptor, a physiological repressor. And what we found is that these molecules, if we gave them to the animals in vivo, so he did all the medicinal farm just to make them bioavailable, they cross the BBB, they don't have toxicity. We've been giving them for 18 months now. We haven't found any major toxicity, but what we find is that they biodistribute very well. So if we use our reporter animal, this is a healthy animal, and if you remember, dots means CMA activity. So you see very nice activation in the liver, for example, but you also see this beautiful activation in neurons, and we also saw activation in the retina. So we thought that we could start using these drugs for some of these age-related diseases, what we have seen a decline in CMA. And the first disease that we did was frontotemporal dementia, just because Mathieu happened to be very interested on that condition. And he has shown, if you remember, that the mouse models of frontotemporal dementia and the patients had this decrease on CMA. So he used one of these aggressive models where you have very early synaptic loss, and then you have the cognitive function and tau pathology. So we gave the drug at the time where you have already synaptic loss, and we look at behavior. So these are the wild type animals. The mouse model is called PS19. It's a model of frontotemporal dementia. They develop this hyperlocomotion, and also they lost the sense of fear. They are in the middle of the field. When we treat with these compounds for 3 months, we already see that the hyperlocomotion decrease, and now they recover this sense of fear. They don't go to the center as often. We also check memory. So the ones with vehicle, they have bad performance on the memory assays. Meanwhile, the ones that are in the drug, they preserve memory. And also depression and anxiety, that are 2 features of this animal, they seem to also be improved. But we wanted to know that this improvement, because we are giving them systemically, we wanted to know that we were doing something on the brain. So the first thing that we did is to look at proteotoxicity. So when you compare, these are 3 regions of the brains. These are wild-type animals. These are the models of frontotemporal dementia. We are staining for tau, for hyperphosphorylated tau. So you can see here this huge accumulation of tau in these models. And the question was, after these 3 months of treatment, have we cleaned up some of this mess? And this is what we found. So for what it's worth, we can eliminate phosphorylated tau in an animal. We can still not do it in people, but I think we are in the right direction, that not only we are improving functionality, but we are doing it in part by restoring proteostasis in the brains of these mouse models. And then, of course, we wanted to do something similar for retinal disorders, and the first one that we did was retinitis pigmentosa, and this was mostly driven by the mechanism of action of the compounds. As I mentioned, retinoic acid receptor is inhibitor of CMA, and what we found is that if you block this receptor, obviously you are going to have more CMA, but you change so many other things. So what we have developed is molecules that only stabilize the interaction of this endogenous repressor anchor with the receptor, and that only affect, instead of the 275 genes that are under this receptor, it only affects 32 genes, and 13 of them are part of the transcriptional network of CMA. So we think it's a very small dedicated program, and we use retinitis pigmentosa because we found that patients with retinitis pigmentosa have very low levels of anchor. So our idea was like, okay, if they have very low levels of anchor, but we can stabilize the little anchor that they have, we might be able to take out this inhibition and activate CMA. So we did that in this mouse model, and as you know, these are very aggressive mouse models, but just by treatment, daily treatment during the early stages of the disease, if you compare the one with the vehicle, has already this thinning of the retina, and you can see the retina here is better preserved, and also the length of the rods. We did electroretinograms, and as you can see here, the animals preserve sight, and I don't have the image here, but we also decrease the level of gliosis that was associated to the disease. So we think again that by enhancing CMA, that is what the animal started to do, but if you remember then it kind of goes down. So if we maintain this enhanced CMA, we can preserve at least a structural function of the retina. And then we also wanted to do it in macular degeneration, and the credit really goes to Patricia and Deb. My contribution here was quite small, and is Iñaki again, the one who did these studies. So we using IPS reprograms from the patients with macular degeneration or healthy controls. If you look at the levels of CMA, as I mentioned before, they have lower levels of CMA, but you gave this drug, in this case it was only 24 hours as a proof of concept, and you get this very nice upregulation of CMA, not only in the healthy retinas, but also in the, not retinas, IPS differentiated in retinal pigmental epithelial cells. They also restore CMA activity almost as close to the healthy individuals. So now that we know that the drug is able to reactivate CMA, can we improve the other properties that we saw in this macular degeneration? So if we look at proteostasis, so if you remember there was a lot of proteotoxicity, and just with the drug you can reduce it. The proteotoxicity is only in the macular degeneration individuals, not in the controls. We look at the maximal respiration because we, as I mentioned, CMA seems to modulate the metabolic switch. And if you look all the individuals, every patient that has been treated, all of them you have an improve in the OCR. We also look at lipid peroxidation that was enhanced in the patients, but if you treat with the compounds, lipid peroxidation decreased, and the same is true for DNA damage. These differentiated cells have very high levels of DNA damage in the patients, but we were able to normalize them with the treatment. So again, these are just only experimental approaches. There are in human primary cells, but we still need to do this, move it into patients. But, it seems to be going in a right direction. And just to summarize a little what I wanted to convey today, I hope I have convinced you that CMA activity is one of these forms of autophagy that decrease with age, that there are sex differences that I think are important to consider, not only quantitative, but also qualitative. And then that, of course, this pathway is very important for quality control. And I showed you the aggregates in the neurons or the presence of proteotoxicity in the retina, so contributes to degeneration. But that part of this phenotype is not only accumulation of garbage, but it's also the loss of regulatory functions. And I talk a little about how CMA contributes to remodeling the presynaptic and postsynaptic proteome, regulation of metabolism, and even regulation of other proteolytic systems such as the proteasome. And then in the last part, I gave you some examples of how we are trained to repair CMA by showing you the effect of the drugs in neurodegenerative conditions and also some of these retinal disorders. So with that, the most important slide, obviously, is the amazing people that I have the luxury to have in the lab. They know about everything. I only know about lysosomes. We have amazing collaborators in all the things that I've been showing, and we are lucky to have people that still support our lab. So thank you so much for your attention, and I will be happy to take any question.
Thank you, Anna. This was truly wonderful. I have a little bit question out of your scope of your presentation. Did you look at the CMA during the development, very early in life?
Yeah, so that's a great question. Should I stop sharing so we can see each other? Or?
Yes, please.
Yeah, at least now I will have this humongous head in your screen. But so we have so something that is very interesting because I've been talking about this LAMP2A. And this is a splice variant. So basically you have LAM2A, LAM2B, and LAM2C. And what we notice is that during development, the splicing changed a lot. And I actually was reported very early on by other groups. So for example, when you look at neurodevelopment in the brain, originally, you have mostly LAM2B that does macro autophagy, and then as the cells differentiate, then you start having much more of LAM2A. So there is this change. So now that has motivated us to start looking at stem cells in the brain. So we have neuronal stem cells and how during neurodifferentiation you change these levels of CMA, and if you don't have CMA, differentiation is biased toward glia. But there are other parts of the lab that have been looking at development in skeletal muscle so far, and brain. Those are the only two organs that we have looked a little at development.
Thank you so much. We have one question from Zoom. Alan? Are you muted, but you're still good looking?
Well, maybe you'll change your mind if you could fix your glasses. Anna, as usual, it's a tour de force, and thank you so much for sharing this wealth of data. It's just spellbinding. I have 3 questions, and maybe I'll start with the last one, which is probably the simpler one. Is how are the sex-related changes in CMA directed?
Yeah, so we think it's a combination. I mean, obviously, there is some hormonal regulation of CMA, so there is some cell extrinsic, but the cool thing is that if we isolate neurons, primary neurons from males and females paps, they already have those differences. So something that I didn't mention is that LAM2A is in the X chromosome, and we thought that there is an inverse relation between CMA activity and the expression of 6, that, is one of these genes that repress the S chromosome. So we think that leakiness of the S chromosomes might be behind that, but of course, at the level of, so that will be quantitatively, right? Like having more or less LAM2A. The interesting thing is like, why different substrates? So for that, what we think is important is post-translational modification. So as you know, for example, acetylation, we did the acetylome, and it's very different in males and females because metabolically, you are very different, right? The amount of acetyl-CoA that you produce is going to be different. And those post-translational modifications in lysines are going to be determinant not only for CMA, but also for proteasome and anything that use a lysine as a marketing. But if you remember the motif, we have a lysine. So if that one gets acetylated, that changes also the landscape of what is being degraded.
Fantastic. So that gets to my more maybe complex question, and if you don't want to bother with it, it's okay. And that is, can you comment on the changes in the relative participation or contribution of the different proteolytic pathways? I mean, you showed so many fabulous, distinctions of CMA in all kinds of conditions. What if you take a look at the big picture? CMA versus macroautophagy versus pathway.
Yeah, that's a great question. And I mean, in part we have been doing, but through the years, I think we all have done it wrong, because what we do to see how much proteasome contributes, you put proteasome inhibitors. Macro, you put inhibitors. As soon as you inhibit one, the others many times can change their contribution, right? Because now you have this rerouting. So the way that we have gone about, and we have done it in parallel, is to use reporters. So we use Degron, we use macroautophagy reporter or CMA. So this way we don't interfere. So what I can say more or less for what we have learned is that it's very cell type dependent, but also context dependent. The same cell that is very dependent on proteasome, if you start doing oxidative stress that damage mitochondria, then macroautophagy is the only one who can save the day. Not CMA or proteasome can degrade mitochondria. So, there are these differences across that. And something that we found is that this mechanism of crosstalk, they seem to also be very cell type dependent, because today the talk was all about like brain and about the neck. We don't see compensation. We eliminate CMA, and proteasome is working, but it doesn't do the extra mile of compensating, and macro the same. If we do it in the liver, then you don't see aggregates, because you block CMA, macroautophagy and the proteasome get upregulated, and that you lose the regulatory function, but you don't lose quality control. So you don't see aggregated, the same thing in the skin, macrophages don't revelos aggregate, but then anything that is more central nervous system, this crosstalk somehow is not there, and we don't know why. So we would love to understand how is the crosstalk in the liver and try to do it in the brain to restore these pathways.
Fantastic. It's just mind-boggling. So my final question had to do with changes. What changes are genetically dictated or environmentally imposed, like in the alteration of protein?
CMA. Yeah. So for CMA, the only thing that we have, because something to keep in mind, because it's a splice variant, many times it doesn't even show in any transcriptomes or any GWAS. I mean, they say LAMP2, but you don't know what LAMP2 is. So for example, LAMP2, the whole gene is affected in Dannon's disease. So of course, because they don't have A, B, and C, part of the problem is because they don't have macroautophagy. Part of the problem is because they don't have CMA. So, they develop neurodegeneration and heart problems. But we cannot say it's only because of LAMP2A, because also B is affected. The only thing that we have is more two things. So we have one study that we did with a group in Spain on patients at risk of cardiovascular diseases, and they identify a SNP that only occurs in LAMP2A on those patients, and it occurs in the LAMP2A exon. So that was interesting, because that means that modification somehow is make them more predisposed. And we have some studies that mouse models with no CMA, they are very predisposed to atherosclerosis. You know how difficult it is to create atherosclerosis in a mouse? You take out CMA, they develop plaques. So we think there is some relation there. And then in the positive sense, in collaboration with Yusin Shah, she was studying centenarians. And we know centenarians have better CMA, at least their fibroblasts and their T cells. So she found that there is not in all the centenarians, but there is a family, like kind of big group of centenarians that seems to have also a variant in the LAMP2A, and this is in the non-coding region. So we think that microRNA regulation might be something that we are missing. So I would love now to have some studies trying to see if microRNAs modulate the splicing or just the expression of LAMP2A. So that's the only thing that we have, just because centenarians have that variation in the non-coding region.
Fantastic. Anna, you may be one of my favorite lecturers.
I don't know that, but you have heard me many times.
Hey, Anna, this is Dorota. Fantastic. I just can say the same as Alan. Wonderful talk and beautiful data. I have a question. What happens to CMA in senescent cells? Because I would say it's actually probably activated. What can you tell me?
Yeah, so we have one of my graduate students, she's defending in 1 month, and we just submitted today her thesis. So the whole thesis is about senescence and CMA. I can give you a full talk. No, but it's very cool actually, because what happened is that we found that most cell types, like when we look at cancer cell, like, cell lines, primary cell, if you engage into senescence, but all kind of stressors. I mean, we have DNA damage, telomere shortening, we have that genetic modulation of TER. They always upregulate CMA when they engage into senescence. So we're like, okay, that's fine. But if you do it in an old cell, you still engage into senescence, but this CMA upregulation doesn't happen. And what she has found in her thesis is that the lack of CMA, so CMA is not required for senescence because you don't have CMA and she can still engage in senescence, but is required for the clearance of senescent cells. So you know how senescent is supposed to be transient. If everything goes okay, you have this transient release of SASP, and then basically macrophages came and NK cells came, and the immune system cleared them up. If you don't have CMA, you engage into senescent, but the composition of the SASP change and become toxic for the immune cells. So you cannot remove the senescent cells, but also the surface proteins that normally are utilized to say the eat me, not eat me, also cannot be retrieved. So basically they are continuously expressing this not eat me signal. So then senescent persists. And as a proof of concept, we use these tracks that I show you that activate CMA in 18-month-old animals. And then we have them for 6 months with the drugs. And when you look at levels of senescent burden in brain, liver, and adipose tissue, they have less amount of senescent cells because now they are being removed more efficiently.
Oh my, thank you.
Yeah.
Hi, Dr. Cuervo. This is Emily. I'm an MD PhD student in Dorota's lab. Thank you so much for this wonderful talk. You mentioned that the decline in CMA with aging is largely due to this LAMP2A receptor on the lysosomes. So what is responsible for this decrease in LAMP2A? And similarly with this compensatory upregulation with CMA that we see, is it dependent on LAMP2A? Is LAMP2A also compensatorily upregulated?
Yeah, so great question, both of them. So if you would have asked me like 2 years ago, I would have said it's all post-transcriptional. We never see changes in expression per se of LAMP2A with age. Cells are still expressing because all the studies that we were doing were in the periphery. So for example, in liver, you see these very low levels of LAMP2A protein as the animals get old, but transcript remain constant. So, and then for a while I thought it's like, oh, maybe it's the trafficking to lysosomes, but no, the protein-rich lysosomes, what we saw is that the protein becomes unstable. So your LAMP2A, like any other protein, right, in your cell, have to turn over. So normally we measure the half-life. So when it reaches the lysosome to be a receptor there, normally it has a half-life of 3 days. So it's a very long-lived protein. And every 3 days you have, you renew it. What we found is that in all animals and also cells from all people, like primary cells, this half-life, instead of being 3 days, is 10 hours. So that means that during 2 days and a half, you are not expressing, you don't have this protein, even you express the same amount, right? It's just because it's being degraded faster. And in the case of the liver and macrophages, we were able to link it to changes in the lipid composition of the lysosomal membrane. So basically, we did lipidomics, and what we found is that the post-translational, phosphorylation of lipids seems to be altered, but also as you age, your lysosomes start having more ceramide, and ceramide start trapping this LAMP2 and make it to be degraded. So basically, we think that maybe if we can get some lipid scavengers, we might be able to restore the normal lipid composition of the lysosome, and then we will not have to overexpress the protein as we do in the animals. But we think that in that case, it's post-transcriptionally. Having said that, we have done now studies in the immune system, like T cells, and also in neurons, and there we can see that there is a translation. So the level of LAMP2A that is being translated seems to change on those cells. And it's because there is an RNA binding protein that sequesters. It increases with age. And it's interesting because it has been shown to have, it's been a risk factor for Alzheimer and Parkinson. And what it does is that it traps the RNA of your LAMP2A. So of course you are transcribing the same, but now part of your RNA is there like trapped in this RNA binding protein, so it cannot be synthesized as a protein, and you have less. So I think the mechanism we are still learning, but it's a little cell type dependent. At the end of the day, these drugs that I presented today, what they do is they transcriptionally upregulate not only LAMP2A, but also 12 other components of this small pathway of LAMP2A. So we think that the drugs are having more effect than just overexpressing LAMP2A, because eventually other components might also change, right? So with the drugs, we keep the stoichiometry, and that seems to be working well.
Thank you, Anna. We still have many, many questions, so
I will go short, very short.
Yeah, Franz Winberg, UCI. So I'm interested about the CMA activator in the retina. So did you look at the retinal ganglion cell hyperactivity? Because I think that is thought to be at least partially induced by the retinoic acid receptor activation.
Yeah, so great question. So I didn't go into the chemistry of how we did this. What we found is that this hyperactivation, so that's depending on the retinoic acid receptor, but not modulated by ENCOR in that case, is mostly depending on the coactivators and another co-repressor. So since we are only touching ENCOR, like the interaction with ENCOR, we didn't see those changes. I was very worried at the beginning touching the retinoic acid receptor because it's also so important for neurodevelopment and the other thing, but this is very different from the antagonists or inhibitors that are out there. This is just acting on the ENCOR axis, and actually the next generation is much even more specific just for that interaction now that we know the surface.
Yeah, I'm Bing from Metamine Silas Lab, and so I have maybe 2 questions. You see the regular people, the health will decrease in the last 10 years. And so is this also happening in the rat or mice when in the lab animal? Because they are everything almost they're the same for the lab animal. do you see this same thing happen in the?
Did you turn on?
Sorry, it's cutting a little. Can somebody repeat the question?
Turn it on now? There's a green light on it.
No. How to turn it on?
Oh,
yeah. Hello, can you hear me better?
Oh, yes, much better. Yeah.
Sorry. I'm Bin from Metadensilas lab. So you report in the regular people, the health will decrease in the last 10 years or a little bit about that. Is this also happen in the lab animal, say rat or mice? Because everything will be controlled almost the same for the lab animal.
That's a good question. So it's very strain dependent. So the steep, how abrupt they start deteriorating depends on the different strains that you use. But it's true that this loss of function occurs way earlier than death. And in fact, that's why we normally use animals at 22, 24 is our aging, because once you pass that, the animals have a very severe, similar to people, they start having more propensity to spontaneous cancers. They have alopecia, they have loss of skeletal muscle. So it also happens gradual, but the animals can live till 32, 34 months old. And that period from 24 to this 30 long is when they have this gradual functional deterioration, very similar to people. But again, the kinetics, very strain dependent.
Hello. Hi, I'm Kim Gokowski. Thank you so much for sharing all of your wonderful work. I am still thinking about your graph where you show the kind of stable level of CMA and then it drops rapidly with time. And then I was thinking, but then, when we're born, it suggests that people who are born, when we're born, you start back with good CMA levels, right? And so kind of similar to Dr. Palchevsky's question, what is it that allows an embryo or an egg to kind of reset their CMA?
Yeah, maybe I confuse everybody. The first graph was health, was not LAMP2.
Oh, got it. Okay. But so it still degrades
So for
over time.
So for LAMP2, look, no, this is a very good question. So now I understand because, so the first one, I was just plotting health of people. So normally you are okay and then you start deteriorating. With LAMP2A in mice, we have seen that till 12 months of age, you have very high levels or whatever are the levels of CMA. And this decline in LAMP2A in particular starts by 12 months. There is only one organ, so the skeletal muscle starts a little earlier, but most organs, we just see it past midlife of the animals. So, and as you say, you reach your levels after development and you keep those ones for almost half of your life. It's just later that it starts deteriorating. And we think it's a combination of all these problems that happen because of metabolism. We can reproduce that same decrease in LAMP2A just by putting young animals in high-fat diet. So these changes in the lipid membrane composition, we think that happen gradually, but they start mostly by midlife.
But so then do you think that children, when they're born by their moms, are they inheriting kind of degraded activity generationally over time? Or is it that women give birth early enough before they demonstrate generation? And that's why fertility is limited to a certain age range?
Yeah, we haven't done anything because it will be very interesting to know, the epigenetic traits are inherited, right? So, but we haven't done anything on that respect. So basically nothing related to pregnancy and inheritance of CMA activity. We just use the single strains that we have.
Hello, can you hear me? Yeah.
Yes.
Amazing talk, Dr. Cuervo. This is Priya from Dr. Zode's lab. So I have two basic short questions. So the first one is, do you see CMA decline as a early driver of age-related diseases, or is it a secondary consequence of cellular stress? Number one. And second question is, how reversible is CMA decline with aging?
Yeah, so great question, both of them. So we think it's an early event based on the evidence that we have so far. So, for example, going back to the skeletal muscle, what we did some studies in humans. So with Luigi Ferrucci has this population or this study that he called the Gestalt, that these are healthy all individuals. So the condition is that they have to maintain, independence, activity. So they are still very healthy. And even at the molecular levels, we start seeing this decline in CMA that then, for example, when they get into sarcopenia, it gets much more accentuated, but it seems to be an early event. And in the case of the animals, which pathway goes down first regarding proteostasis is also very organ dependent. So for example, there are in some tissues that we know that LAMP2A is the first one to go, there is compensation, and then the others go. But there are, for example, tissues like in retina, surprisingly enough, macroautophagy is the first one to go, and CMA is holding the fort for a while, and then it eventually goes down. So it might just be very, depending on how this crosstalk that we still don't know enough is in each of these organs. But we think at the molecular level is an early event, and actually we are now, for some of the, we are looking at brains of individuals that they haven't developed Alzheimer's yet, but they just die for other things, and you see a very good correlation between presence of hyperphosphorylated tau. Even they don't have a full bloom symptoms, they already have these changes in LAMP2A and CMA. And then regarding the reversibility, that's something that we always wanted to do, because I was okay, we can prevent, right? Like we do this mouse model that now express these beautiful levels of LAMP2A, and they don't develop, or they live longer. I didn't show them today, but they live longer, they are healthier. But we kind of wanted to use the drugs for that. It's like, if it's now down, can we revert? Can we restore? And what we have done is, like, the latest that we have given these drugs is 22 months old, so that's quite old for an animal, and we still get, it's interesting because you get restoration of CMA activity. And when you look at each of the components, it's not that all of them go back to when you are young, but having these levels, even they are lower, but because they increase over what you had before and they do it stoichiometrically, they seem to improve. So we have done that, for example, for senescent. We have done that for muscle. So we did it for the animals, by 18 months, and then they recover muscle force. And then we also did it actually for hematopoietic stem cells of all people. So as you know, stemness is lost with age. So we were doing some study with a collaborator in hematopoietic stem cells. So we got them from young people and old people. And if old people that already have very lower repopulating ability, if we treat for like 1 week with these drugs, they definitely restore CMA. We measure that, but they also seem to gain repopulating activity. What we don't know if there is a point of no return. What will be the point that we cannot bring this CMA back? So far, as I said, the latest that we have done in animals is 22 months.
Hello. Dominic Lewandowski here, Kaiser Lab. I have a quick question. You mentioned ceramides that they can inhibit LAMP2 and CMA. So I was wondering, because in RD10 there was this quite severe accumulation of ceramides, or also in your model, do you see any rescue or normalization of the lipid composition and ceramide composition in the lysosomes after you rescue CMA?
I love that question, because we got the data. The problem is that we didn't put it in the paper because we couldn't get it at the lysosomal level. So what we did the treatment with the drugs, and then we did lipidomics of the whole cell, because isolating lysosomes, that we can do in liver, but you need such a large amount of cells that we cannot do that from the amount of cells in cultures that we have, or from the retinas. But for what it's worth, when they did the lipidomic, we can still, we see I will say normalization. I don't know how to describe, but levels of ceramide decrease, and then you have like these more healthy lipids. And then, by staining, the H&E staining, it also seems like there is less lipid peroxidation, for what is worth. But I cannot conclude that this is happening also at the lysosomal membrane. We know that levels of LAMP2A are higher, so I think it's probably because they get stabilized, but we couldn't isolate lysosomes.
Thank you. All right, Anna, we have one more question. We could keep you for another hour or two. That's absolutely true. And by the way, I love your decoration. So you're living with CMA in your office.
Of course.
Every day, every moment of the time. So let's finish with Kim's question. And again, thank you so much for being with us today. Kim.
Thank you for having me.
I'm Kim. I'm from Dr. Prochaski's lab. I have one question that how would you comment on the high-fat diet and a high-protein diet related to the CMV level?
So we know that we did these studies that we put the animals, young, healthy animals, we just put them in high-fat diet, and we monitor changes in CMA. So it turns out that the first 2 weeks, in high-fat diet, they upregulate CMA many organs, like, liver, of course, pancreas, but also we saw even upregulation in skeletal muscle and in other organs. But then once you pass the third week, you have this inhibitory effect. And in that case, so we did I think it was 8 weeks. And when we isolate the lysosomes and we compare the lipid composition of those lysosomes, because this was in liver, with the one of an old animal, they have many resemblance. And we also show that in that case, you destabilize LAMP2A. So that was for the high-fat diet. The other thing that we have done is high carbohydrate diets. So what we found is that in high carbohydrate diets, so these CMA deficient animals, the ones that are deficient in the whole body, they seem very sensitive to these high carbohydrate diets. They become very obese, they develop insulin resistance like there is no tomorrow. So we were very intrigued. So what we did was to follow, if we put them in a high carbohydrate diet, what happened, and then this initial increase of CMA only lasts for 2 days, and then it goes down. And we've been talking a lot, and it's great that Alan is here, about the possibility of glycation of LAMP2A in these high carbohydrate diets, because we found that, for example, with aging, we have found glycation of HSE70, but we have not been able to determine if LAMP2A also gets glycated. So that will give us an idea of why CMA will decrease on those conditions. We haven't done anything with high protein diet so far.
Thank you again. Please join me to thank our speaker.
No, thank you for having me.